Simon, thank you so much for joining me today. You and I have worked together for almost 2 decades now, and
Mm,
it's really amazing what you've been doing as an investigator. You're a professor of neurosurgery at Duke
hmm
University and a member of the Duke Molecular Physiology Institute, where your Translational Basic Science Lab is located. Simon, I'd love for to start with you talking a little bit about the work that you did on the Human Genome Project. How did that experience shape you as a scientist and what you do today?
Um, it was a fantastic experience. I think quite often when you're that close to history, you don't realize that you're making it. And certainly I think I was a research tech when I first started. I was the 53rd member of the Sanger Institute in the UK, which sequenced a third of the human genome. And I think there's probably 1,500 individuals there now. So it was very, very early in the Sanger's development. And because I was there early, I was able to see the Human Genome Project sort of develop as a large, collaborative, coordinated sort of effort where everyone pulled together towards a common goal. And I think it was, we had some interesting times. We had the emergence of Solera and Craig Venter into the competitive space, which was interesting both scientifically and politically. But it really galvanized those of us who were working on the Human Genome Project, and it was thousands of people involved in it. And so I think it had a lasting impression upon me with respect to the value of team science, the value of sort of flat administrative structures where everyone felt engaged in science, and sort of really framed my perspective of how science should be carried out going forward.
Simon, maybe I can follow up on that because you really have been, while leading your own, great lab, you've been a very collaborative scientist.
Mm.
Within Duke, you and I have worked together on cardiovascular
hmm
disease together, but also outside of Duke. Can you talk a little bit about how you view team science and collaborations?
Yeah, absolutely. I think one of the main things that I'm sort of known for, and we can get on to talk about single cell and spatial, and people just assume that I work on just direct the core, but my own science has been heavily involved in multiple sclerosis and then Alzheimer's disease, autism, and then brain tumors more recently. And it's pretty apparent that we can't carry out this sort of science as basic scientists from my own perspective without the engagement with physician scientists, without clinicians, without statisticians, bioinformaticians, etc. And so for my own neurological disease research, those collaborations have just been fantastic for pushing the science forward. Um, and again, it's sort of this team science approach. With respect to the core, I don't see the point in developing or instituting technologies that don't benefit everyone. There's no point in being siloed away with respect to technology. I think it's, the rising tide sort of analogy benefits all those who are carrying out disparately related science, but the technology may be really important for other people you just haven't thought about.
Yeah, Simon, and I think what you bring to the table, right, as a molecular geneticist, a basic scientist, but also somebody who understands, even if you're not a clinician, you understand the clinical questions that need to be
Mm,
asked. For
mm,
example, in your multiple sclerosis work, I think it's really fantastic that you have this diverse expertise, but then you pull in teams so that you can really exponentiate science. And I think it's a perfect model of how we need to think about genetics, genomics moving forward so that we
mm.
have the largest impact on human health. So just kudos to you for being able to do that. On
Thanks.
that note, can you talk a little bit about your core? You're really one of the world's experts on single cell, single nuclei technologies. Can you talk a little bit about the work that you do in the core and how you collaborate in that space?
Yeah, sure. So I've always been a bit of a tech junkie. So when I was at the Sanger, myself and a colleague who's now at the Jackson Labs developed a method of uh, fluorescently mapping or constructing physical maps of genomes. It had been done radioactively until that point. So we transitioned that radioactive technique into a higher throughput fluorescent technique. So that was used on a fairly large proportion of the human genome. And I think ever since then, I was interested in sort of novel technologies and how they can be applied more broadly to science. I think if I think it's a good idea for my own neurological disease research, I feel like other people might be interested in it. And so it's a matter of seeing what technologies are really transformative. Identify where there's an opportunity with these novel technologies, how we can change the paradigm from approaches that we've been using traditionally for a long time. Apply these technologies that give us a unique aspect on the science and be able to understand the mechanisms of science. And I think that's why, we were one of the first labs on the East Coast, I think, to get into single cell with 10x Genomics. We also maintain that relationship with 10x by adopting spatial transcriptomics approaches. So not just looking at very high resolution single cell, but putting it in a spatial context. And then we were the fifth group in the world to have 10x's Xenium platform, which is their in situ sequencing platform. And then we most recently were the first independent use of the platform in understanding diffuse midline glioma. So it's sort of this transition of getting from where we used to be doing particular approaches to understand science to really adopt these novel technologies to ask novel questions.
That's great. It's really a fantastic core that you run. And I think you've done a lot of really important work across many diseases, including your focus in brain cancer. As we finish up, Simon, I'd love to understand, kind of your vision. What do you think the future of genomics will look like?
Well, that's either a loaded question or a transformative question. I think it has to be multimodal. I think, we have to look at the translation of, instead of unimodal approaches to understanding the mechanisms associated with disease development and progression, we have to have this sort of multi-omic approach. My move to neurosurgery most recently coincided with me starting a program in neurosurgery. It's the Brain Tumor Omics program. So what we're trying to do with BTOP is really, as I said, integrate these multi-omic modalities. So if you think of the genomics, transcriptomics, epigenomics, proteomics, metabolomics as being these horizontal omic modalities, and we want to vertically integrate them across cohorts to try and use the power of each of these omic modalities to have a more informed understanding of disease development and progression, and just sort of make not just novel discoveries, but translational discoveries where we can identify potential novel therapies, the impact of different treatments in the context of brain tumors, but then also integrate modalities that we haven't thought of before, such as digital pathology, AI means of diagnostics, empower the diagnostician, if that's the word, so the pathologists or the clinicians at the coalface of interacting with patients, understand better the diagnosis of disease development and progression. So I'm really, really excited by what omics and genomics specifically are going to deliver in the next 3 to 5 years. I think it's going to be a very rapid improvement in what we can do.
No, that's great. I think the power of big data, both on the imaging clinical side, as well as what you do on the omics side, and pairing those through team science, right, is how we're going to really be able to personalize how we take care of patients.
Absolutely. Yep.
Simon, it was so great to talk to you today. I
Pleasure.
really appreciate your time, and congratulations on all the fantastic work that you're doing.
Thank you very much.